Welcome to our presentation on the second United Nations Sustainable Development Goal, Zero Hunger. This goal aims to end hunger, achieve food security, and improve nutrition and promote sustainable agriculture. We chose to focus on Target 2.4, which aims to, by 2030, ensure sustainable food production systems and implement resilient agricultural practices that increase productivity and production, and that strengthen capacity for adaptation to climate change, extreme weather, drought, flooding, and other disasters. This target is important because sustainable food production is the first step towards zero hunger. Sustainable food production is necessary to establish food security and ensure a population's health and productivity, therefore putting barriers in place to prevent them from a continual cycle of hunger and poverty. There is only one indicator for Target 2.4, the proportion of agricultural area under productive and sustainable agriculture. Within this, there are 11 sub-indicators. We will be looking at 1. Productivity, and 10. The prevalence of moderate or severe food insecurity. Let's examine the current situation. Indicator 1. Since 2018, the global percentage crop yield has dropped by 5%, down to 56% in 2020, and kilogram yield per hectare has increased only slightly. Looking at sub-indicator 10, food insecurity has also worsened, increasing from 21% in 2014 to 30% in 2020. As it stands now, 1 in 10 people are suffering from hunger, and 1 in 3 have moderate to severe food insecurity. This is bad news for global sustainability, and an integrated effort must be made to keep us on track to achieve the goal of zero hunger by 2030.
One of the barriers to achieving this goal is climate change. Average global temperatures are rising, and extreme weather events are occurring more frequently, for longer, and with greater severity. Agriculture is the most vulnerable sector to this rapid climate change. Of particular concern is the increasing severity of drought, which affects crop productivity in about a third of the world's population. Over 300 million people in Africa rely on the corn for food and income. However, corn is particularly vulnerable to drought, with crop losses ranging from 30 to 90%, leading to hunger and poverty. One response to this is the use of genome editing technology, known as CRISPR, to modify corn to make it drought resistant. First, scientists identify the DNA they want to remove or alter. This is the target DNA. They then create guide RNA, which is complementary to this target DNA sequence, allowing it to bond specifically to the target. The Cas9 enzyme cuts the DNA at this location. DNA can be removed or inserted, and the cell will naturally repair the cut, resulting in a new, modified piece of DNA. Scientists have used CRISPR-Cas9 to inhibit stress signals in corn plants, allowing them to flourish despite adverse conditions. This particular modified corn saw yields of 2.5% or 5 extra bushels per acre every year compared to normal corn when growing in drought. CRISPR has also been used to create other drought-tolerant and high-yield crops such as wheat, tomato, and rice, and is being used to increase crop resilience to other stresses such as soil salinity, flooding, and disease. When modifications are compounded, larger increase in yield may result. Moreover, CRISPR-modified crops could deliver even higher yield if current obstacles were addressed.
With the current factors limiting the use of CRISPR-modifying crops being difficulty regenerating the modified cells, the number of known target genes, and a difficulty delivering CRISPR to cells. To improve this, modified cells should be grown in a variety of different growth media and conditions to determine what best promotes regeneration. Comparative genome analysis should also be conducted to aid the discovery of more potential target genes in crop plants. Evaluating the indicators of Target 2.4, CRISPR-Cas9 modified crops are showing promising increases in yield, improving Indicator 1, productivity, and, since they are more resilient to adverse conditions, it will allow for more consistent food production, improving Indicator 10, food security. CRISPR-Cas9 modifying crops directly address the aims of Target 2.4, as it is a resilient agricultural practice that increases productivity and production and strengthens capacity for adaptation to climate change, making it a promising part of current and future sustainable food production systems. This makes the possibility of achieving zero hunger in maybe even our lifetime possible. There are many challenges with CRISPR though, as it currently stands, but this just means that the future for biologists going into genetics is a potential gold mine, and they'll need all the help they can get.